I'm Delanne Elliott Midland. I'm the president and CEO of the End Brain Cancer Initiative. I want to welcome all of you today to the new developments in PCNSL treatment webinar. Let's introduce Cameron Lilly. Cameron is with Belay Diagnostics. And I'm really excited today to hear from Cameron. When we get to listen and learn from him, I think all of us are going to go, wow, if I can find out this or that about my disease without a biopsy, right, or surgery, how interesting and evolved is that, right? And so we're going to learn a little more about that. Cameron earned his PhD in molecular virology, and was then introduced to the challenges of rare diseases and cancers during his postdoctoral research on oncolytic virotherapy. From his postdoc, he entered industry to leverage his passion for strong science and science communication in sales roles at Cell Signaling Technology, where he developed his commercial acumen. From there, the clear patient need led him to joining the liquid biopsy space, first as a scientific affair liaison, then as an MSL for cell-based and cell-free technologies for monitoring and characterizing cancer. He brings his 5+ years of experience in liquid biopsy and interfacing in the healthcare space with healthcare providers and key opinion leaders to the team at Belay, where he has joined as MSL to bring his passion for cutting-edge and patient-centered medical science to the challenging field of CNS tumors. Cameron, thank you for joining us today.
My pleasure, Delanne. Thank you so much. It is a joy and an honor to be here and to have this be the first of our partnership conversations with brain cancer at Belay Diagnostics. This is a really momentous occasion for us, and we are very excited.
That's what partnership does, right?
It is
We
amazing.
get to create excellent patient disease education and patient access.
Absolutely. And that's part of what I hope to talk about today. So at Belay Diagnostics, we are constantly striving to reach new heights for CNS testing, and our company focuses on what is called liquid biopsy, so looking at genomic characterization and epigenomic characterization of tumor-derived nucleic acids from cerebrospinal fluid, specifically in the arena of CNS malignancies. Belay Diagnostics is very unique. We are a fairly young company, but that does not mean that what our company does is new by any means. We are built on more than 10 years of research at Johns Hopkins in premier labs, and we are purpose-built from the ground up for utility in cerebrospinal fluid. This is a key delineator for us, and it has allowed us to work out the kinks and challenges of CSF-based genomic testing. We also are very unique in that we are extremely science-minded. We are the peers of peer review, and all of our tests are thoroughly validated, and our validation is peer-reviewed and published. Our flagship product, Summit 2.0, has an analytical and clinical validation that is peer-reviewed, and we also, in that peer-reviewed validation, were able to assess our test performance in CSF that was tissue biopsy matched for clear, true positive, clear, true negatives. And so we have very, very strong assays on the market that are backed by tremendous research thanks to the quality of our exceptional lab staff. As I mentioned, we're a little bit of a young company, officially founded in 2022, and we launched onto the market in January of 2025, bringing our initial offering of Summit, which was a genomic profile for CNS tumors, and Vantage, which looks at methylation of the MGMT promoter region. This is also important for CNS cancers. What we learned very quickly after coming to the market was that our initial offering of Summit was too limited. And so in less than a 10-month time, we were able to develop and release with a full clinical and analytical validation, Summit 2.0, which interrogated more than 10 times as many genes and included the addition of a number of other biomarkers which were not a part of Summit. It was a tremendous endeavor and something of which we are extremely proud. In the first 12 months, or now almost 14 months since our commercialization, we have put out 12 peer-reviewed publications, which of course include our validation studies. We have 5 published case reports and a meta-analysis on CSF-based next-generation sequencing testing. And what I'll be telling you about today is the story of the largest CNS study lymphoma performed by any commercial partner, which is a 12-month retrospective of the experience of using our Summit test in CNS lymphomas. And so that will be the meat of my talk today. Something that has been wonderful about coming to the market in this area where there is such a great need with the challenges of CNS cancers is that we have found fabulous partners to work with in the field to help bring our testing into clinics for patients. We're now in over 120 institutions and more than 80% of NCCN designated cancer centers, and we work with more than 250 providers at this point now. And featured here is just a short list of some of the truly prestigious and world-class organizations that we're working with to try to help support their patients. So, as we've talked about a little bit before, we know that central nervous system lymphoma is a rare disease, and the rates are greater in the elderly population. This accounts for between 4 to 6% of new diagnoses of malignant brain cancers. And therapy is challenging. As Dr. Nayak went through, a lot of the treatments that are effective in non-CNS lymphomas do not appropriately treat CNS lymphomas. The diagnostic pathway we saw before always begins with brain and spinal imaging. Cerebrospinal fluid testing is a part of guideline-based diagnosis of CNS lymphomas. This includes CSF cytology and flow cytometry to look at the presence and characterization of malignant cells, to look at gene rearrangements of a gene called IgH. MIDI88, which is a key marker in this, can be assayed by PCR, but this is also one of the 4 arenas in CNS malignancies in which CSF-based tumor DNA testing and genomic characterization is actually recommended by the guidelines themselves, and that's where we at Belay Diagnostics play. Of course, if at all possible, a biopsy is performed, and as was mentioned by Dr. Nayak, an ocular exam is extremely important as eye and CNS involvement tend to go hand in hand in these diseases. So we know that there are challenges in this diagnosis. MRI and CSF have limited sensitivity. CSF cytology in particular has very limited positivity rates and requires repeated performance in order to bring that sensitivity rate up high. And MRI can be deconvoluted by very talented radiologists, but some diseases can look like one another on MRI. For example, GBM or glioblastoma and CNS lymphomas can sometimes present similarly on imaging. Also, brain biopsy is not always feasible. This is an invasive procedure, and depending on the location of the lesion, it may not be possible or it may be too risky to go in to look for a biopsy. Well, what about testing in the blood? This is common in solid tumors and has become an industry standard in many diseases, but for central nervous system malignancies, blood-based testing has not really borne out due to the fact that very little of the nucleic acid that comes from within the brain and spine makes it into the blood because of the blood-brain barrier. And so that's where CSF-based testing really can shine, and part of what set the stage for a recently published publication. This is from just last month. That's how new this is. And this is the experience and impact of Belay Summit 1.0 in real-world setting, in real patients, in real clinics across the US on CNS lymphoma. Let's talk a little bit about Summit 1.0. This was the initial version of our offering and has since been expanded upon greatly in Summit 2.0. And Summit 1.0 was for looking at both gene variants and chromosomal alterations from tumor-derived DNA in cerebrospinal fluid for informing diagnosis and management of CNS malignancies across the spectrum. It was a 32-gene panel that assayed 112 hotspots across those 32 genes and also looked at, of course, chromosome copy number alterations by looking at whole arm losses and gains. This is a key hallmark and biomarker for human cancers in general. Now, for CNS lymphoma, the 2 major genes on the platform that we think about are, of course, MIDI88. This is, as Dr. Nayak said, a key biomarker in this, and detection of the L265P variant in that gene is diagnostic for CNS lymphoma, but also key in this is looking at CD79B. Now, I'll mention one of the other tests that we offer. This was a first to market for anybody. We were the first people to bring CSF-based testing for MGMT promoter methylation to the market. This is great because it's, of course, less invasive. And our testing is extremely unique in two ways, one of which is that we use an enzymatic conversion for methylated DNA bases rather than a chemical conversion, which allows us to retain that precious input tumor nucleic acid. And so we don't destroy half of our template in trying to analyze it. We also can turn this around in very quick time because of the qPCR-based assay. It requires very low input DNA. Now, while this is recommended for testing in all confirmed or suspected high-grade gliomas, MGMT promoter methylation is an emerging biomarker in CNS lymphomas. And so it was exciting to see that in the field, in our retrospective analysis, MGMT promoter methylation was analyzed as a way to both help understand diagnosis and the disease as it stood. Workflow for Summit 1.0 is quite straightforward. We take CSF. We actually only need a single tube of cerebrospinal fluid to perform our assays. That remains true. Isolated nucleic acid, and this is where the molecular magic that we exclusively license from Johns Hopkins for use in CSF comes into play. It actually allows us to build the analytical, what we call libraries for understanding what's happening on the genetics of the tumor from the very same input strands for both our aneuploidy analyses and for Summit, but also to retain those original input strands to look at methylation on those. This is extremely powerful technique that allows for the sort of multiplexing of looking at these biomarkers and is the backbone of the Summit and Vantage workflow from the initial period of our commercialization. So the question was, how is Summit really helping in CNS lymphoma in the clinic, in the real world, with real patients and real clinicians? So we wanted to assess that, look at how it was performing in confirmed and suspected CNS lymphoma, both primary and secondary, and also to see, can we quantify or understand the impact of the results of the test? Beyond, was the test positive and negative? How is it really helping? Is it really helping? And so we were able to look at a cohort of 48 patients corresponding to 50 specimens because 2 of our patients actually were tested before and then subsequently to therapy. So we can look at how therapy impacted their results from a 12-month period. In this cohort, 37 patients had Summit included as part of their initial diagnostic workup, and they were submitted to us with concerns that ran the spectrum of CNS cancers from we suspect primary CNS lymphoma or secondary CNS lymphoma to try to resolve whether it was primary or secondary to determine if it was primary or secondary CNS lymphoma with the potential for other CNS malignancies. But even some were much more general, submitted to us with provisional diagnoses of just abnormal brain scans or potential for ocular lymphoma. Is it malignant? Is it not malignant? We had one patient in this cohort, which was actually confirmed to have CNS lymphoma, that was submitted with a provisional DX of high-grade glioma. And so that highlights the importance of getting this kind of molecular testing. There were also 13 patients that had previously been diagnosed with CNS lymphoma. So those diagnoses were both primary and secondary CNS lymphoma, ocular lymphoma with CNS involvement, and a more general CNS lymphoma diagnosis as well. So let's dive into the data. How did we do? So in 64% of our tests, so 32 of the 50 total, we were able to detect clinically relevant genetic variants. These can be both single nucleotide variants or insertions and deletions, that plus aneuploidy. Aneuploidy on its own, so these are again the loss or gains of entire arms of your chromosomes, which is a key marker of cancer, and in some cases, methylation of the MGMT promoter. Now, you might think that 64% detection rate might be a little low, but remember we're talking about impact, and I'd like you to keep that in the back of your mind as we progress through the talk, because there is value sometimes in a negative result, and we had quite a few cases for which negatives were still very clinically impactful. What did we find? What gene variants in particular? I'll highlight the top 3 for us. Of course, one of the most common that we found is the MYD88L265P variant. This is absolutely canonical for CNS lymphomas, primary and secondary, but especially in primary. And this is not found in other CNS cancers. And so this marker by itself can be considered diagnostic and clear for CNS lymphoma. One thing we found that we were a little surprised to find was that multiple variants in a major tumor suppressor gene called TP53 were found. The protein encoded by TP53 is a master regulator of cell cycle and is commonly associated with cancers, and it's associated with a poorer prognosis if your tumors are TP53 variant positive. We did also, of course, find multiple variants in CD79B as well, and this is included as part of the diagnostic workup per the guidelines in this space. Let's talk a little bit more about what are some of the other things that we found. So gene level variants are a key part of Summit and the remaining key part of Summit 2.0. But one thing I think is underappreciated generally in our field is the power of the detection of aneuploidy as a biomarker for human cancer, especially in cerebrospinal fluid. This can be done and has been done and is well established in the literature to be an extremely sensitive way to detect CNS cancer. And in 5 of the cases for which the provisional diagnosis testing was submitted to us for secondary CNS lymphoma, there were no gene level variants detected in these patients. However, 5 or more chromosomal arm level losses or gains were found in these patients, which informs a diagnosis of CNS lymphoma. The presence of this level of aneuploidy absolutely corresponds to, or at least is heavily suggestive of, the presence of malignancy in the central nervous system. And so even when gene level variants are not detected, testing like ours can help to find the signals of cancer by using sensitive methods to detect aneuploidy. Some other ways in which our testing was very impactful in patients for either primary and secondary CNS lymphoma was detecting gene level variants when the clinical concern was for progression. And so being able to detect these helps to support the idea that using molecular characterization and highly sensitive methods like ours, we can find the molecular signals and evidence of progression or recurrence and use that as part of the clinical toolkit to help make these diagnoses, to help inform care for patients. And so there were a number of these that were included. Now, remember when I asked you to keep in mind the value of a negative result. There were a number of cases for both initial workup and in the group that had previous diagnoses for which negative tests, i.e., testing for which no genetic variants were detected, were very important for informing the diagnostic pathway for those patients. In the top group, you can see that there were 5 patients for which CNS lymphoma was part of the differential diagnosis and was one of the many potential disease states that could be happening in these patients. And in those patients where it was negative, we were able to tell those providers that at least based on our testing, this does not look like a malignant origin for what you're seeing. It's always nice to be able to tell someone, hey, it looks like you don't have cancer, at least as far as we can tell. Now, I'll couch that very closely. We are not a standalone diagnostic. We are part of the toolkit, but this is nice information to be able to give a care team. Say, hey, based on what we're finding, we think that there's likely a different etiology for what's happening in your patient. In the previously diagnosed group, we find some very interesting results. 4 patients were actually submitted for testing after having received therapy, 2 of which had been tested with Summit 1.0 prior to the application of therapy, and the lack of variance in that instance would correlate well with a response to therapy. We actually just recently published a paper not a week and a half ago that describes how looking at the molecular signatures, which we call variant allele frequency, can actually inform a molecular signature for a response to therapy or not. And so this was great evidence that suggested testing like ours may actually be able to help inform and confirm that there was indeed a response to therapy when taken into consideration with other clinical correlates. We're actually able to speak a little bit more to one of those as we have quite a bit of post-testing follow-up information for this particular case. The one patient here was study ID 4 and study ID 7. This patient had concerns for vitreoretinal lymphoma, had a vitrectomy to confirm that there was indeed ocular lymphoma here. And of course, the concern was always, is there CNS involvement? Our initial testing on that patient came back with MIDI88 L265P as detected, which informs that there is indeed CNS involvement in this patient. And with that information in hand, the care team went with systemic methotrexate instead of local intraocular injections. The subsequent testing on this patient revealed that the MIDI88 L265P variant that had been present in the initial testing had been reduced to beyond the level of detection. And that correlated with clinical response to therapy as well. So again, this shows how monitoring disease and monitoring response to therapy can be done using CSF-based genetic testing. In this particular case, we're looking at a very drastic reduction. This patient had variants in CD79B, MIDI88, L265P, and two different variants in TP53. And then after therapy, this patient had no detectable tumor-associated variants by Summit 1.0. Again, bolstering the case that this kind of testing can be used to understand and assess response to therapy. So let's talk about impact, because that's one of the major facets of this, is beyond just the data. Did it actually change how the clinicians were thinking about disease? Did they impact therapy? Did they impact differential? And we did have a few cases for which, as we say in the field, they were lost to follow-up. And so we were unable to determine the impact of our testing in those. However, I think that the real key take-home here is that if you look across the spectrum of challenges that these providers were looking to try to address utilizing our testing, in 82% of cases, the results that we provided, be they positive or negative, led to a clinically significant impact on that patient. And I think this is something to keep in mind as we move forward, because it bolsters that, yes, this is a fairly good sized study, but it's retrospective. So now this sets the foundation to continue to evaluate the utility of tools like ours. So, what have we learned? One, CSF-based tumor DNA testing is a very powerful technique. It's extremely useful. And in the study presented today, we showed both the ability to detect gene-level variants in relevant and important genes for CNS lymphoma and chromosome-level arm-level losses and gains. Summit 1.0 was capable of giving that information in both primary and secondary CNS lymphoma, and the expansion of Summit to Summit 2.0, combined with the results of this study, I think really firmly lay the foundation for promising and exciting future directions for the utility of precision diagnostics like ours in CNS lymphoma. I'll leave you with a little bit of a description of where we are now. I mentioned that that was a retrospective study looking at the first sort of 12 months of our offering. Our initial Summit testing was quite limited compared to what we have now. So we now have 3 tests on the market, Summit 2.0, Ascent, and Vantage, that are designed to be utilized as you see here. Our comprehensive genomic profiling via Summit 2.0, which pairs with Ascent, assays 520 genes. It looks at copy number variation for 62 genes, agnostic or partner agnostic fusion detection in 28 genes, as well as key biomarkers in cancer like tumor mutational burden and MSI. And Ascent is now where our chromosome arm level loss and gain testing lives. We've also added the capacity to look at the basically amplification or copy number loss of discrete small areas of chromosomes called focal losses and gains. And that is a highly sensitive way to bolster our copy number variation genomic profiling results across the spectrum in CNS cancer care, including in CNS lymphoma. Vantage is, of course, included there. This remains unchanged so far, but we will see where methylation takes us in the future. We're always trying to strive to be better at the things that we do. And so with that, I'd like to thank you so much for the opportunity to speak with you and to introduce myself and Belay Diagnostics and our testing with you. Thank you for your attention. And what questions do you have?
Thank you, Cameron. That was amazing. I have a question for you.
Shoot.
As you know, the End Brain Cancer Initiative is educating patients, families, caregivers, care partners, and actually other health HCPs on these tests. And you know that we have patients working with Shreya on that, et cetera, et cetera. But pretend that's not happening. Just pretend for this webinar's sake, I'm a patient. I'm a PCNSL patient, right?
Mm-hmm.
Okay, well, I want to know one, why didn't I get this test or both of them and as appropriate? And two, how can I move forward to receive these diagnostics?
Absolutely. Very happy to answer that question. So, one of the reasons why a patient may not have received our testing at the time of their diagnosis is because we are still a fairly young company. We've only been commercialized for a little bit over a year. And so, if your diagnosis happened prior to that, this technology was simply not available. What I would absolutely say for anybody that is interested in learning more about this or trying to obtain our testing for you would be you can either contact us directly or have your provider contact us. And the contact information on the screen goes to our amazing customer service representatives who I make no exaggerations whatsoever, are incredible, and they work day in and day out with providers and patients. We can work with you and your care team to figure out how to make it such that you can submit a CSF sample to us for comprehensive genomic profiling. We're very excited to do so, so we encourage you or to have you encourage your provider to get in touch with us. If you would like to, you can also reach out to me directly. I am always happy to field questions and to try to put people in touch with members of our organization who can help get that process started. We're always here to try to get our testing into the hands of as many patients as possible, because that's really the primary mission statement of our company, is to serve patients and those who care for them. So that's what we want to do. So please get in touch with us if you can. We're here to help.
Excellent. Thank you. One comment I was thinking about on your workflow slide for these diagnostics, and you mentioned it. One tube of CNS fluid is required, not two, correct?
Correct.
Okay.
That's absolutely correct. And that remains the case even with the increase of our panel size, even with all of the additional biomarkers that we have added in Summit 2.0 compared to the original offering. We still can run all of our testing, of course, assuming that there's sufficient nucleic acid content, on one single tube of cerebrospinal fluid of 6 ml.
So on that slide, if I'm thinking from a patient voice, patient perspective, that kind of thing, I would just put one tube.
That's a good idea.
Just because we've had lots of conversations with patients. Yeah. Okay.
100%.
Yeah, it's a scary thing to some patients. And so less can be more.
Oh, absolutely. And it's never fun to think about sitting down for a lumbar puncture. You're talking about something that, even though it is less invasive than something like resection or a stereotactic biopsy, you're still having a needle inserted into your spine. So it's a little hairy. And so we definitely want to make sure that, and part of the reason why being able to use one tube is so nice is that there are other parts of the diagnostic workup that are run on CSF. And we want to be included as a part of that, as part of the toolkit for providers.
Absolutely. And so I'm also thinking about the patient population, family care partner population that will go forward to see this particular webinar and this patient disease education video. Would you please take a moment and explain further? You referenced insertions, deletions of nucleotides. Would you just take a moment to explain that in patient-friendly terms?
I will do my best. Okay, so there are a couple of different kinds of, we tend to refer to them as variants. You may hear mutations that can occur. This can occur at the changing of a single letter of your genetic code, which would be called a single nucleotide variant. Sometimes multiple of those can be changed at once. That would be called a multinucleotide variant. Or there can be very small sort of sequences that are stuck in where they shouldn't be or have been deleted when they really need to be there. And that would be what we would call an insertion deletion or an indel. And so our goal is to be able to test for as many different kinds of what we would largely call like genetic insults or variants, to make sure that we can get the best classification that we can for the genetic information that we have, because different variants may have different implications for the impact on the protein encoded by that gene. And so being able to assay many different kinds allows us to have great information to provide care teams about what to do with it. What kind of variant is it? Does it cause a loss of function? Does it cause a gain of function? Does it cause that protein to just stop when it should be much longer? That's called a truncation. And we can do a lot of that with the genetic testing that we offer.
Outstanding. Thank you.
There's one comment. I do love circulating tumor DNA and CSF for diagnosis. There's one caveat, and I think patients need to know about that. If there's a large tumor in the middle of your brain, the only thing you never do is a spinal tap. And so that is the only limitation, and that's what we have done in our trials where we collected spinal fluid. We sometimes you just can't do a spinal tap because it's just not safe for a patient. But I'm a friend of
absolutely
collection. But even if the test exists and even if your provider can order the test, there might be a reason why it's not done, because it's just unsafe to collect the CSF.
Sure.
Yeah.
And we would never ever want to or even implicate our testing in any situation where it might not be safe.
Yeah. Just as
we'll also make the note that we can accept cerebrospinal fluid collected by any methodology, not just a spinal tap.
Oh.
So that can be collected from an Ommaya reservoir, that can be collected from a VP shunt. It can also be collected during craniotomy in surgery if you're so inclined. But yes, absolutely, Dr. Gomes, you're 100% correct. The idea is always to do what is going to be best for the patient. Yeah. And we would never even want to be considered in an instance where it would risk the patient or present any harm. Absolutely correct. But that's a great caveat. Appreciate that.
That is unfortunate caveat. Yes, that is true.
It's a challenging field, and these are very challenging diseases. Absolutely.
Cameron, I had a question. So from the diagnostic standpoint, what are some of the most common delays or missed steps you may have seen in getting a PCNSL diagnosis confirmed? And then how can tools that Belay Diagnostics have can help close that gap?
So I would probably defer to the clinical colleagues that are on the call with me to see what kind of diagnostic challenges that they find, as I am not a diagnostician. But some of the things that I outlined in the beginning of the talk are challenges in this area. And that is that on imaging, CNS lymphoma can sometimes look like other CNS malignancies. And the limitations of some of the standard tools for CSF analysis, especially cytology, are there. It has a very limited ability to be sensitive. I will say that the diagnostics have gotten much, much better, and the diagnosticians have been getting much, much better. But there are cases in which, biopsy is infeasible, in which the best way to get at the genetic information that would really aid in nailing down that diagnosis might be from CSF. And that would be where our testing really comes in, is to be able to get to that molecular characterization when otherwise it might not be available.
Thank you.
You're welcome.
Thank you, Cameron, so very much.
Pleasure.
Really good information. Really appreciate it. Thank you.